Hi, everyone. Thanks for attending another one of our WISC STEMinars. Today we have Tyler Purdue. Tyler is a graduate student at Columbia University in the Biological Sciences Department. And today he'll be talking to us about genetic engineering. And before you start, Tyler, we have questions for you. So did you always see yourself ending up in a STEM graduate program?
No, I didn't. I took kind of like general biology in undergrad at the University of California, Santa Barbara. Just knew that I was like interested in it, didn't really know exactly what I wanted to do with it. But grad school wasn't really on my radar at that time. So I went into work for various biotech companies after graduating. And then after working for like 5 years or so, I kind of wanted to like get to the next level. So I decided to come back and pursue a PhD.
Nice. And what is like your most favorite part about doing your PhD so far?
I think it's pretty cool just how much control you have over what you're doing. Like you pretty much decide like what you want to work on for the next few years or so. And yeah, it's basically like no one's really telling you what you have to do. You just kind of have that autonomy.
That's awesome. All right, thanks. Yeah. So thanks, Tyler. And anyone, you should completely feel free to unmute yourself if you have a question for Tyler. And if you don't want to, feel free to type it in the chat and I'll read it out for you. So whenever you're ready, Tyler, thanks.
Great, thanks. Yeah, so today I'll go over genetic engineering, basically what it is, kind of the basic premises behind it, like how you actually do this, and then also just some cool examples of genetically engineered or modified organisms that have already been created, and then what end up at the end with kind of what the future of genetic engineering looks like, specifically in humans. So first, we should start out just by defining what genetic engineering is. And it is the direct manipulation of DNA of an organism to change the phenotype in a particular way. If you're not familiar with the term phenotype, there's kind of like two principles in biology. There's the genotype, which is the genetic makeup of an individual, basically like the DNA code that an individual has. And then the phenotype are the physical traits or characteristics that are coded by that genotype or that genetic material. So an example of that shown here is eye color. So you have different genes that code for different eye color. That's the genotype. And then that results in the phenotype of the actual color of someone's eyes. So genetic engineering is a very recent advancement in history. We didn't even know that DNA and genes were the mechanisms of hereditary till the mid-20th century. And direct modification of genes wasn't developed until the 1970s. So this is all really recent technology and it's still advancing pretty quickly. However, it's worth noting that humans have been influencing the genetics of organisms for a very long time. People have been doing this genetic manipulation for thousands of years, even though they were unaware of the specific genetic mechanisms underlying that. So shown here are the results of selective breeding or artificial selection for certain traits. Here we have the ancestor of the dogs that we all know and love. So you have the wolf here, and then by selecting for certain traits like height or length of nose or even personality traits like loyalty or aggression even in some dogs, you end up with two dramatically different looking animals shown here with the pug and Great Dane. And then also...
So we have a quick question actually from Amidou, and they ask, so is there a machine to put the new genotype into a person?
Sorry, can you say that one more time?
Yeah. They ask, so is there a machine to put the new genotype into a person?
Is there a machine?
Yeah, like I guess they're asking you from your previous slide. Like, could you put any new genotype or maybe modify? Maybe they're asking the genotype in a human.
Yeah, so I'll go into that at the end. Like I'll show you like how this is done, what the tools are, and then talk about that. But yes, there are, and it has been done already. So yeah, I'll talk about this. So shown here is the example of the dogs, and then also you can look at what this kind of genetic manipulation through selective breeding has resulted in with what we have today, corn and watermelon. So thousands of years ago, corn was just like a very small grain, and then by breeding for the traits that we want, like bigger corn kernels, sweeter taste, this is what it looks like today. Same thing with watermelon. Used to be like a very small kind of seed-like, and then now today we have like the super sweet, really big watermelon. So I just want to go into just kind of some basic information on what DNA is, what it looks like, and then some definition terms that are useful to know, especially for the rest of this talk. So DNA codes the information to make proteins, and it's heritable. Therefore, it has two jobs. The first is to determine amino acid sequence. So amino acids are the subunits that make up proteins, shown here on the right. So we have the DNA here. It goes through one copy into RNA. We won't really go into detail on that. And then that RNA gets made into protein. And these individual circles here with the letters in the middle, those are the individual amino acid subunits. And then the second job of DNA is to determine its own sequence. So it directs the synthesis of its own copies. So that way, that DNA or that genetic material can get passed on to the next generations, and then those next generations can have the same DNA to make those same proteins. Proteins themselves are kind of large, complex molecules that play many critical roles in the body. And those proteins are what result in the different phenotypes that you see in an organism. Just some additional terms that are useful to know. So when I reference them throughout the talk, you know what they are. The first is a gene, which is just a DNA segment that contributes to a certain phenotype or function. So this can kind of be thought of as, most of the time, a gene is just a specific part of the DNA that codes for one protein. And then the genome is just the complete set of genes or the complete DNA sequence of an organism. So then here, just going to show kind of what DNA looks like and what the structure of it is. So I'm sure you're all familiar with this double helix model shown up here on the top left, where you have a sugar phosphate backbone with bases spanning across the center. And those bases are in DNA. They're either adenine, thymine, guanine, or cytosine. And those are what determines the DNA sequence and then what proteins get coded off of that. So this is typical of what you'll see on the bottom right here of a DNA sequence. It just lists the sequence of the one strand, and then all of the A, T, Gs, or Cs in that sequence in the right order. Okay, next slide. So you can't just add DNA in solution with a bunch of cells, and that DNA will get uptaken by the cells, because all cells have cell membranes, and then additionally some have cell walls also protecting the cells. So that way, you need a method to get that into the cells. And this kind of goes back to that question we had before, and like, is there a machine, or like, what's the way that you do this? So there's a handful of different ways that have been developed to get the DNA into cells. The first is using viruses. So DNA is put into a virus. That virus then infects the cells and delivers the DNA that was put into that virus. That DNA then gets uptaken by the cell and then incorporated into the genome of that cell. Another one is microinjection. So this is where they literally take like a very, very small needle and just inject the DNA into a cell. This is limited to very large, like mostly eukaryotic cells. Then another really common one that's used in like microbiology labs is electroporation. So for that, you apply a weak electric current to the cells, and then that forms little holes or pores in the membrane that the DNA can pass through. Very similar to that is chemical pouration, and that's just where you put the cells in a solution of different chemicals that also produce little pores in the membrane that the DNA can pass through. Then there's laser pouration, where tiny laser beams create those holes in the membrane. Gene scissors are a technology that uses lasers to cut like whole genes and then insert those into the cells. Gene guns are kind of futuristic or sci-fi, where they literally take DNA in metal slivers, and then just it's like this big machine, and then they fire the DNA into cells. This one's also limited to large cells, especially like plant cells specifically. And then finally, there's oh, sorry, Ariana, go ahead.
Yeah, so you're sending like tiny shrapnel into like a cell culture? That's wild.
Yeah.
That is so, I don't never heard of that. Does the metal just like stay there and the cells heal away from it?
I mean, I'm not super familiar with the technology. I haven't used it myself because I don't work with plants. But yeah, I think I'm not entirely sure how it works. But imagine like the metal has to go somewhere. I don't think it just sticks around. It would probably mess up the
Cool, thanks.
cells, yeah. And then finally is nanoparticles. And that's where DNA is coated in certain particles that can bind to the membrane and then pass through that membrane, and then the DNA gets into the cells that way. So yeah, there's a bunch of different technologies to do this. I would say kind of the main ones that are used are the viruses, electroporation, chemical poration, and then the nanoparticles. Those are the ones that I've been exposed to the most, like with the genetic engineering that I've done. Specifically for DNA or genetic engineering in people, going back to that question before, the main ones are the viral particles and then nanoparticles, because obviously you can't like shock a person and get the cells in get the DNA into them that way. Okay, so now we know how to get the DNA into the cells, but where does this DNA come from? Like, how do we get the DNA that we want to add to these organisms? So there's two main ways to do that. One is to amplify it from an organism's already DNA sequence. And then the second way is just to synthesize that particular sequence in the lab. So the way to get it from a sequence that's already in an organism, so if you have a certain gene, like in a bacteria, that you want to take and put into a plant or something like that, in order to get that specific DNA sequence from that bacteria, you would use this method called polymerase chain reaction, or PCR. And this is used to amplify a specific region of DNA and to get a large concentration or many, many copies of that sequence. And those many, many copies are needed to do further manipulation. You can't just have one copy of a DNA sequence and really do anything with it. So I'll briefly go over what this method looks like. So you have your original double-stranded DNA, and you want this one particular sequence right here. So you design these DNA primers, which are just single-strand DNA. And you design these so that they bind to the outside of the specific sequence that you want. And then by going through different temperatures and using an enzyme or a protein called DNA polymerase, DNA polymerase amplifies across those primers to give you the sequence that you want. So with multiple cycles of changing the temperature to denature the double strand, anneal the primers to those single strands, and then amplify across that sequence that you want, you're able to get millions and millions of copies of that particular sequence in just a couple or a few hours. So this is a really, really powerful tool. And it's used basically for every single thing in molecular biology and especially genetic engineering. And then the second way to get a DNA sequence is to just synthesize it in the lab. And this is actually how you get these primers that you want to amplify a certain segment here. So without this synthesizing, you wouldn't even be able to do this PCR reaction. So just a brief overview of how do you synthesize DNA. Essentially, you have a starting molecule with the base that you want to start with. And then again, they just do add different chemicals to remove. So it starts out with this block on the end. And then by adding different chemicals, you can remove that block. Then you add like the next base that you want in your sequence. That gets added with the block on. Then you remove that block again, add the next sequence. So you can build these specific sequences that you want in the lab.
Oh, Tyler, I have a question. Sorry. I got a direct message, which I answered, but I think it was meant for everyone. It says, I did an experiment once when I took the DNA out of a strawberry. I used soap, isopropyl alcohol, and strawberries. Which way of DNA delivering was used in this experiment? And I think maybe making the distinction between the delivery and the lysis is.
Yeah, so that was, in that one, you weren't delivering the DNA because you were just taking it out of the strawberry. You weren't like putting it into the strawberry. So in that case, that's DNA extraction. So DNA extraction is basically with those different solutions that you added, you're just lysing the cells. And then when the cells lyse or break open, that DNA gets released. And then you can purify that DNA from that lyse solution. Yeah, so that's kind of the first step before you do this PCR reaction, right? You need to isolate or extract the DNA from the organism that you're taking it from.
Yeah. And we do something really similar in lab, right? As the dish soap and isopropanol, right?
Yeah, definitely.
Awesome, thank you.
I also got a direct message by accident. So they're asking, how long do each of these processes take in general?
They're really quick. So again, I guess in total, try and break this down. So like DNA extraction, to get the DNA to amplify, you can do that in like an hour or two. And then ordering primers, designing the primers, you just design those on your computer based off the sequence that's known. That can take like 10, 20 minutes. You order those through a company that then synthesizes them. This is really quick. So normally you like order these primers, they get synthesized and then delivered to you the very next day. So now you have your primers. Then you can run your PCR reaction, which takes, depending on how big the fragment is that you want to amplify, it usually takes like 2 hours, I'd say on average. So yeah, it's all really quick.
For anyone who doesn't know, could you explain why you need a primer?
Yeah, so the primer determines the sequence that you're amplifying. So you have your one on the right side and then one on the left side. And those kind of set the bounds of the sequence that you're amplifying across.
Awesome, thanks.
Okay, so now we know how to get the DNA, how to get it into the cell. But if you just add DNA into a cell, it won't do anything. First, you need to make a break in the DNA, in the genome that you want to incorporate. Specifically in this, the part that you want to insert into. And that can be done in a few different ways. The first is with restriction enzymes. So these are proteins that cut in very specific sequences. And they cut in this same place in every sequence every single time. So the first, like shown in this example here, is this enzyme or this protein called EcoR1. And this recognizes the sequence GAATTC and then cuts between the G and the A on those. When it cuts, it results in this sequence right here with sticky ends. And then if you have DNA with those same sticky ends on the end, or that same sequence, then the cell's natural machinery will repair that cut. And so these proteins cut in the same place every single time. So you can only really insert in that one section. But there's a couple of other technologies that have been developed that allow you to pick and choose any sequence in the DNA to cut and then insert your DNA into. There's 3 of them shown here. The first is zinc finger nucleases. The second is transcription activator-like effector nucleases, or TALEN. And then the third is CRISPR-Cas9. I'm sure you've probably heard of CRISPR-Cas9. It was developed fairly recently, and it's been a huge breakthrough in the genetic engineering field. And basically, all these allow you to program a particular enzyme or a protein to cut in the place where you want to insert your DNA. And then again, the cell's natural machinery will then take the DNA that has the same ends, or like matching sequences on the ends, and then insert it into that specific site. And that can be done in, there's 2 different cell machineries, I guess, that do that. The first is non-homologous end joining. And that just kind of like pushes the ends together. I won't go into too much detail on this. And you can see with that, like this purple segment right here, if you have, this is the gene that you want to enter into the DNA. So by breaking it apart and then having the sequence that matches the ends on both sides of that break, then you'll insert the DNA right there. And then the other one is homologous recombination. And then again, this works in a very similar way, where if you add your donor DNA sample with the matching ends, then it'll get inserted into the DNA in that specific spot. All right. So that's kind of how it's done. And then now I'll go into just some cool ways that this has been used and that you'll see like in everyday life, this technology has been used. So the first is, I'll go over like ways in bacteria and yeast. So the insulin that people inject if they're diabetic used to be obtained from pig or cows. And now they've made a bacteria and they inserted the gene to produce insulin. And now you can just grow up that bacteria in a big tank, harvest it, and then collect the insulin from that, purify that, and then that's what diabetics use today. Another example I'm shown here is in a yeast cell. And this was actually from a company that I used to work for. And they engineered yeast to make artemisinic acid, which is an anti-malarial drug. So they inserted 22 different genes or the DNA to code for 22 different proteins. And then with that, you can just feed these yeast cells sugar, and then they'll produce this anti-malarial drug for you. So this has also been done in plants. Shown here on the top left is for golden rice. Golden rice has been modified to produce vitamin A, so that people in areas that don't have easy access to food with vitamin A won't develop a deficiency for that. And this was actually one of the very first engineered crops that has been developed with genetic engineering. They took 3 different genes. 2 of them came from daffodils, and then 1 of them came from a bacteria. They then introduced that into the rice, and then that makes the rice produce vitamin A. And then shown here is kind of the color difference. So vitamin A has an orange color to it. So you can see the rice here are orange or golden. That's where the name comes from. Another one is BT cotton. And BT cotton has been engineered to produce an insecticide to combat a specific worm called a bollworm. That gene that got inserted into the cotton came from a bacteria called Bacillus thuringiensis, whereas that's where that BT comes from. It's from that bacteria's name. Essentially, the cotton produced this toxin where if the larvae for those worms eat the cotton, then they'll die. And this is beneficial because you don't have to spray these crops with like kind of like broad use pesticides, where they just naturally produce this, and then that way you don't have to spray the crops that could have other detrimental effects to the environment or to other animals around. So you can also genetically engineer animals. Shown here are 2 examples of that. On the left here is, on the bottom left, this is the structure for a protein called green fluorescent protein. And this was actually discovered by one of the professors here at Columbia, and he won a Nobel Prize for that discovery. This protein comes from jellyfish. So they found the sequence, the DNA sequence for that gene, for that protein. And then you can introduce that into various animals. Shown here is an axolotl that has been engineered to produce this protein, and then it glows bright green when you shine a black light on it. You can also buy like fish at the pet store I've seen. I forget what they're called, but they have like green fluorescent protein or red fluorescent protein, and you can get those there. And then shown on the right here is goats that have been engineered to produce spider silk. So they took a gene from the orb weaver spider to produce spider silk, put that into a goat, and then that goat produces spider silk in the milk. So you can just harvest the milk, isolate that spider silk, and then like that way you're able to obtain large quantities of that. And spider silk is like one of the strongest materials in the world. So it has many different applications that you can use it for. Okay, this is kind of the last slide. And I'll talk about like how genetic engineering can be used in humans, the potentials, and then also some of the ethical implications behind that. So genetic engineering has a huge potential to treat and cure human diseases like cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS. That's because the genetic mutations are known for these specific diseases. So if you could go in and just add a new piece of DNA that corrects that genetic mutations, you'd be able to cure the disease. So there aren't currently, there hasn't been developed one that's like approved by the FDA to treat a certain disease in humans. But with the CRISPR-Cas9 system specifically that we talked about before, it should be just right around the corner that we start seeing some of these gene therapies, is what they're called, where essentially you do like genetic engineering in humans to treat a specific disease. So there's two ways that you can genetically engineer in people. One is called germline gene therapy, and the other is somatic gene therapy. So in germline gene therapy, you actually do genetic engineering on a human embryo. And since you're doing this on just like the embryo, where there's just a handful of cells, when that cell divides, that DNA sequence will be present or changed in every single cell throughout the person. The other way you can do genetic engineering in a person is to just do it on the person when they're fully developed. So where you target specific cells in that person to engineer. And that one's called somatic gene therapy. So obviously, like the first thing that we want to use this for is to treat specific diseases. But you can also see where this could be kind of applied to other less important things, like changing a baby's eye color or their height or their hair color or their intelligence or anything like that. So that's kind of what the fear of is going forward, is that we're going to have these designer babies where essentially like you pick the traits that you want your baby to have. And if we know that DNA sequence that codes for those specific traits, you can make those changes. So that has some major ethical concerns that people are worried about as this technology progresses, that kind of we as a society will have to place certain limits and certain restrictions in the future as this becomes more of a possibility. So far, only somatic gene therapy has been kind of approved for development, or for kind of these drug companies are able to only try and go after the somatic gene therapy. So only doing genetic engineering on like a fully developed human and not on the embryo itself. However, in the news a year or two ago, you may have seen that a scientist in China used genetic engineering on 2 twin embryos. And that wasn't approved by like the scientific community or any type of overseeing body. So that was kind of frowned upon, but it would turn out to be successful, at least from the data that scientist showed. So you can see that it's really a possibility, and it's developing really quickly. So we kind of have to figure out what the ethics are going forward. With that, yeah, thanks everyone for listening. Just shown here is the laboratory that I'm in with my professor, Dr. Wang, and then everyone else in my lab. And then we have some resident axolotls in our lab. They're just kind of our pets, so they're shown on there.
Thank you.
Thanks, Tyler.
Yeah. Are there any questions? Feel free to ask away.
Yeah, I can share the link to the article about the scientist who did the twin experiments.
Yeah.
I'm still amazed at the goat spider silk.
I remember it was like back when I was in elementary or middle school when they did that, and it kind of blew my mind at the time.
Yeah.
That's amazing.
Amazing.
Yeah.
I can't be comfortable for the goats. For me, no.
I think it's all just like solubilized, so then I don't think it really affects them. Yeah, really purify and isolate it after.
Awesome. So, oh shoot, what was I going to ask? Oh, sorry. No, everyone else go ahead first. We have one in the chat. Let's see. So if you're changing the genes of an animal, would that change how that animal survive? Great question.
Mm.
Yeah, definitely. yeah, that's kind of one of the uses. So I guess going back, let's see, kind of, so this BT cotton right here, I guess this changes, it's not an animal, but this changes how this cotton or this plant survives, because it can no longer be eaten by these worms. Because if the worms eat it, they die. So that's a way that increases the survivability. Actually, a lot of the times when you introduce these new genes into an organism, it actually decreases the fitness, or like the fitness, I guess, like the survivability of that organism. So shown here, like this yeast cell, when you add all of these extra genes and kind of force this yeast cell to produce this molecule that it's not like, nor it doesn't really benefit the yeast cell at all to produce this. It actually decreases the survivability, or like makes it grow slower. Or it's just a little bit sicker. So yeah, it just depends on the genes that you insert in there.
And that's a big question for the babies who were edited most recently as well. Like scientists are wondering like what that edit could have affected in terms of their survivability, and like how it might have affected their immune system and such.
Exactly. Yeah, I didn't go into much details, but essentially that scientist who edited those babies, he made a specific mutation that makes them not able to contract AIDS, which the kind of the problems with that is that there's other ways to prevent that, obviously. And there's also unknown effects that could hurt those babies by making those changes. Because this isn't that well studied and we don't really know all the effects. Specifically that CRISPR-Cas9 protein or enzyme that cuts the DNA doesn't always cut it at the exact place that you want it to. So it has what they call off-target effects. So it'll cut other places in the genome and can add additional mutations that could be detrimental to that person. Any other questions?
I have a question. Do you know which genetic disorders are sort of top of the list in being actually approved, or like probably the ones that'll get approved first for the somatic therapies?
I don't. I'm not really up to date on like the current gene therapies in development, or like which ones are in testing with the FDA right now. But I think those ones that I mentioned, like cystic fibrosis, those are the diseases that like it's just a very short mutation that's well understood and well known. We're just making that one change can treat that illness. Yeah.
We have a question from Anushka. They're asking, I was wondering if there is any way someone could do DNA extraction at home.
Yeah, definitely. I could look it up really quick. I'm sure there's an easy way to do it. Like it sounded like with strawberries, you can just use dish soap and some alcohol, and that extracts it. I don't know, Sergio or Aubriana, do you know the?
Yeah, we have an audience member who's done it before, actually, Emma. And so there are YouTube tutorials that you can find that go through the combination of dish soap and isopropyl alcohol that you can use. So that would be enough to disrupt that membrane outside of the cell and sort of break it open and allow the DNA to like spill out into your solution, and you can collect and concentrate that. It's easiest with fruit like fruit have big cells and lots of loose DNA. I would not recommend it on your own cells. But yeah, there are definitely ways you can extract DNA, and it looks sort of like this. It kind of looks like spider silk almost, or it looks like gooey white strings by eye even.
It's very gooey.
Mm-hmm. Yeah, it looks kind of gross, but it's fun to do. Yeah, just be safe. Always wear gloves. Awesome.
All right, if that's the end of the questions.
Yeah.
All right, thanks everyone. Thanks for taking your Saturday morning. Have a good rest of your weekend.
Yeah, thanks so much everyone for being here. And thank you, Tyler.
Yeah, I guess if anyone has any questions that they think of in the future, I don't know, Aubrey and Sergio, you can give me my contact info. Be happy to speak.
Thanks.
Awesome. Thanks everyone.
Thanks everyone for coming.